Perfect. Well, thank you so much for joining us this afternoon. I am recording this, and we have about 10 people on Zoom and about 8 people in person. And we do have with us Dr. Kyndt from Bellevue University, and you should see him, or you should be able to see him once he starts talking.
Okay.
So I'll let him go ahead and get started.
Okay, I guess I'll talk to the owl. Good. So, well, thank you for inviting me, first of all. I didn't prepare any slides or much for this because I didn't know how formal and informal this setting was. But I can tell you a little bit about the project that we've been doing and how it got started as well. I live pretty close by here. I live in the Leawood West area, so just across Pacific here. So when I run from my house and I come running this way and I run around the bridge and go back to my house, that's exactly 5K. So, so I planned it out to make my run as part of this. So my background, first of all, I'm a biochemist that does microbiology. I'll give you the whole spiel. I'm from Belgium originally, from Europe. I moved to Arizona after my PhD in 2000, and I get the dates now. 2003, I moved to Arizona. I became a research professor there and then moved to Bellevue here to become a teaching professor here at Bellevue University since 2012. So the research in microbiology, on the one hand, I do algae for biofuels, and I study algae and grow them up for the lipids for the biofuels. The other part, I just sample at places, and interesting places where I sample, I try to get to extremophiles from hot springs, from the salt marshes near Lincoln as well. I sample there. And we try to find what's in there, what bacteria are there, and how they cycle nutrients in those environments. And they make good student projects because we find new species in there, and the students can identify those and sequence the genomes, and to get a little project on that. So this started as a student project where I saw these ponds on the creek here as I came running, and I was like, well, what's in there? So I started taking samples, and the very first sample we took was from April. It's in this paper. I think it's in April 2020 when we first sampled. And we just did a 60ness analysis, 60ness RNA analysis, basically looking what bacteria are all in there, and can we get an idea of how unique this is or how different this is. And I wanted to see are those ponds different from the creek that's running through it, and what's the difference between the two as well. Because one is flowing, the other is more stagnant, so there's definitely a difference there. And also looking at the history a little bit of Hell Creek, so we're just talking about this. The thing I found was that the name from Hell Creek came from these farmers that were mad at the creek because it kept flooding every year and it ruined their crops, and they called it Hell Creek because of that. It was in one of the UNL publications, I can't remember which one, but I can find it. But the flooding, I was also wondering, do any contaminants get into the creek and the waters? Do we have any pathogens to come in? Does any runoff get in there as well? So that's really what we're looking for. We only published this first analysis in 2020, I think. Yeah, 2020. Just given as a baseline, saying this is what we found in April 2020. We didn't find any pathogens, anything that was from fecal matter or anything like that. So it looked pretty natural environment, both the creek and the one pond that we measured there. But it was a fun project, so we continued sampling. We sampled throughout the year. We want to see are there seasonal changes in these in the creek and in the pond as well. And now we have data from 5 samplings throughout that year, 2020. And we've done both 60ness ribosomal RNA. I don't know if you're familiar with that, but that's just amplifying a fragment of every bacterium. And then you say, okay, this fragment is present in all of the bacteria. It's like a signature. We can tell, like, this is from Bacillus, this is from Streptococcus, this is from this. And there's thousands of them in all these samples, but you only get a fragment. So now we also did whole genome sequencing, where we tried to sequence as much of all the genomes as we can. It's a lot more data, but then you can get more in depth with that. You can find out not just which bacteria are there, but what do they do? What nutrients do they cycle? Do they take up nitrates? Do they give off sulfur and oxidize sulfur, for example? So we can start looking at these pathways now. Plus, the other thing you can do is you can find viruses in there, and then you can look at the level of how many viruses are there. And most of the viruses we found are bacterial viruses. So bacteria also have viruses that attack them. They're called bacteriophages. They call them phages for bacteria. They call them viruses for animals and plants. But they're essentially doing similar things between these species. We found a lot of bacteriophages, so the phages that go with those bacteria that we find in there. We found some interesting, we're still analyzing those. My colleague, Dr. Moore, who's the plant person, I was talking to somebody about this. He's also a virologist, his background. So he's looking into the viruses and found some interesting things as far as bird viruses are in there and some rodent type viruses that we find in there, probably from birds and rodents passing by, mainly in the pond is what we looked at. So passing by in the pond and leaving particles, leaving feces in there and stuff. So you do pick all these things up, and they do change by season as well. And that's what we're looking at now. How much do they change by season? Because it's quite a change from the spring to the summer to the winter as far as what bacteria are there, and also what viruses are there in those environments as well. So that's all forthcoming. We're doing a lot of analysis still. The two of us, together with some students that come and go on these projects, I'm trying to make a picture of how it seasonally fluctuates in there as well. Some of the things we found, so from these initial samples, we were able to isolate a new species. It's a Rhodopseudomonas species, which may not mean much to you guys, but it's a photosynthetic species that captures carbon and actually grows photosynthetically. And it's unique from any other Rhodopseudomonas that we've seen. So we purified it partially, we sequenced the genome, and now we're fully characterizing that species, and we'll publish that separately as a new species. So we're still thinking of a name of that one too. We got to come up with a name. So something related to Hell Creek probably we'll come up with that. So that's ongoing. Yeah, what else was I saying? Talk about the viruses, about the new species, and I do want to continue the next year and take samples again the next year and see if there's the same pattern that we see. Oh, the other one, I think, for the winter sample 2020, we found that 2 or 3 species were basically making this different from what we saw before. And one of them, this is all the pond area there, one of them was Legionella bacteria. And Legionella causes Legionnaires disease, as you may know before, causes a type of pneumonia that can be caused. So that's the first time we saw, and it's not uncommon in the environment to find this, but it's the first time we saw a potential human pathogen that kind of comes up to higher amounts in that pond as well. I'm still curious where that come from and how that change came about for that winter sample. I want to follow up on if that goes back away and if that fluctuates as well. That's why I reached out to the architects that set this up as well to find out. There's a drainage pipe that comes in there in that pond. And I was wondering, does it come from some waste plant? Or does it come from collection of rainwater? So is it a mixture of the two? And it turns out it's only storm drainage. It's only from the rainwater. There's no mixture with the wastewater coming in at all, which is good. But I'm still curious about that Legionella bacteria, for example, that's in there. Why is that coming up in certain seasons? And where is it coming from as well? Those things. So those questions we still have to answer, but the more data we collect and the more that we analyze this data, we'll try to see patterns, but how does that fluctuate and change with it as well. So that's the stuff we play with. It's really a fun project for us, kind of to see what's happening and how do things change and what can the students learn from these environments, how you have not one bacterium in there, but you have thousands of bacteria in there, how they interact with each other. And it's an easy place for us to sample as well. So there's not a larger application for it. It's really kind of a fun project that we do to see how are things changing and how these interacting with each other in this environment as well. Yeah. I think that's the whole project. Any questions? Yeah.
Just, it used to be a golf course.
Okay.
And I'm just wondering if there's a lot of runoff of chemicals that would potentially be used in keeping the grounds.
Right. I haven't, so we did look at some of the nitrates in there, but that's about all we looked at. We didn't look at sulfur or phosphorus in our analysis. The pH doesn't fluctuate much at all, and the nitrates are pretty stable going through. There is quite a bit of bacterial activity in there, so probably means that the nitrates are fairly high to feed them, and the phosphates as well. But I don't know of any chemicals that are running out that are causing a lot of problems with it. It seems pretty clean for the most part. Yeah.
That is good news. We've been
Yeah.
really worried about this.
Yeah.
There's so much plastic and metal and styrofoam.
There is a lot of trash coming. I noticed that too. And I didn't look at those things. I didn't look at the chemicals from plastics, for example. That's a different analysis that we do, or that we could do. So we didn't look really at that, how that changes to the season. But I noticed that there's lots of particles in there.
Yeah.
As well.
Well, those big culverts are just open.
Yeah, it all comes
So-
running in there.
Yeah. So I don't think that's the only way it gets in there. I think people.
I think
just
so. Right,
Cavalier about it.
right, yeah,
Trash, so,
yeah, I
yeah.
think so.
that's-
But overall, I would
The
say,
salt level isn't high, there's a lot of, so that's kind of normal.
yeah, it's normal, but it's not that salty, yeah, it's pretty, because we'll compare it to other salt areas, like the Nebraska salt marshes is extremely high there, and then we have samples from other places that are salt environments. The species we found are adapted to freshwater environments, so they're not necessarily high.
Have you sampled any of the sediment on the bottom?
We did not. So we took samples from the middle of the pond and from the edge of the pond, so we compared. And the way we typically do it is from the bridge, we lower the device with a rope that samples about 1 or 2 meters deep into the pond, so not much deeper than that. So we haven't gone to the sediment, no.
Is there things in the sediment? This
Yeah.
hasn't been a golf course for 100 years. So
Well, okay. Yeah.
it's not
Yeah, really
stuff
seems to
to
do. Do you know how deep the pond is? I have never analyzed it. So.
Do you know how deep the pond is? No.
Okay.
No, but we've had a couple cleanups.
Okay.
And we have discouraged people from wading out into it because
Yeah.
for one thing,
Yeah.
the water can be so deep, but I don't know how
Yeah.
deep the muck is.
Right.
So
Yeah.
I don't want people to get stuck or
Yeah.
whatever out there. I could just see
Right,
them,
right.
you know,
Absolutely. Yeah.
panicking and
Yeah.
whatever, but, so we
Yeah.
don't have any idea, but, and we
Yeah.
also don't want to be more aggressive with our cleanups until we know what needs to be done.
Right.
So,
Yeah.
maybe you feel like this is frivolous, but for us, it's kind of reassuring to know that the water isn't completely toxic.
Yeah. Yeah.
So,
I said the only thing I was worried about was that in that winter, there was a December sampling that we found of Legionella bacteria that came in there. So I would not go swimming in that environment necessarily. So
December.
yeah, in December.
Yeah.
Not that you have the
Oh,
urge to
God.
do it, but
So have you followed the stream upstream, you know, where it goes, where it starts at and then to that?
I have, so only on the map I looked at it, and I think there's a Boystown reservoir where it collects into, and it comes from one of the Boystown reservoirs, continues the Hell Creek in there. But I haven't physically followed that or take samples there.
So it comes from there and it runs that.
It seems to be running from one of the Boystown reservoirs. And that's where,
Whatever Boys Town used to do, I mean, that could be a source.
yeah,
a lot.
and that's what I was wondering about the creek too, because that's the creek that connects to it. The pond is not connected to the creek, but it has those effluent that comes in there. So, and the effluent just seems to come in that pond from a larger area around here, that just the rainwater comes in there. But I was looking at the creek to see anything from the reservoir, because
Did you sample that? Maybe that'd be good to see.
yeah, it would be good to sample to compare how it changes possibly from the reservoir to the creek. But we haven't done that.
And do you have any idea what this new bacteria does or functions?
It's one of the purple non-sulfur photosynthetic bacteria. So their biggest function is really to capture carbon. They capture some nitrates, but they don't, not a sulfur bacterium, so they don't oxidize any sulfur. It's not part of the sulfur cycle really. It's really carbon capturing that occurs. And they can grow anaerobic and aerobic as well, but they prefer anaerobic. So typically when you have these waterways, you're gonna have algae on top, and the algae growth, they produce, they like to be in the oxygen and stuff, and they produce oxygen. So they're on the top part, and underneath, you're gonna have these photosynthetic bacteria, typically the purple non-sulfurous, because they can tolerate oxygen, but they don't necessarily use it. If you go deeper down, you get the sulfur bacteria, like the green sulfurs or the purple sulfurs. And they don't like oxygen, they like to be in an anoxic environment. So the majority that we found were, depending on the season, in spring and summer, there were more algae on the top. And then we did find quite a few photosynthetic bacteria underneath there, but not too many of the sulfur bacteria underneath there at all. So overall, it seems pretty well balanced as far as algae growing, and then carbon capture and nitrogen capture by the photosynthetic bacteria underneath, and
Would
not
there
too
be
many
any
sulfur ones.
potential for harvesting algae and using it as an amendment to the garden?
I thought you were gonna say biofuels, because that's my other project I work on. I'm not sure what the type of algae is that's in there, because we didn't analyze that part. We just saw that there were algae in there, and we look at the abundance of algae, but we haven't isolated the species from there. They're good at capturing nitrogen phosphates,
Right,
so
so
there's gonna
that would
be,
be
could be
useful,
like a
and then
fertilizer for
carbon.
the, right, could be extra carbon and nitrogen phosphorus for the garden as well. So if you find an efficient way to harvest it, an easy way to harvest it, then you could use it as a fertilizer, that's true. Because what we typically do with the algae is we harvest them for the oils, and then from the oils we make biodiesel, but the leftover algae stuff, you can use as a fertilizer because it's full of nitrates as
So you
well.
have a little unit, we'll take the oil out, sell that.
Ideally,
and then we'll get
yeah.
other stuff and use it in the garden.
Yeah, the
Well, we
problem.
can use the oil to, run a generator.
Right, you could. If it's a species that makes a lot of oil, and that's the question, I haven't looked at that for these species. Because there's about 65,000 species of algae out there, and a fraction of that is good at making oils, and those you can use for the biodiesel. The majority are gonna make starches instead of oils, and those you could use for fermentation and make, bioethanol or something from it, but typically people are after the ones that make the oil, because then you can make the biodiesel from it. So it would depend on that species, what's there, so I haven't looked at that yet.
And then Dr. Kent, I
Yeah.
think Kathy had a question.
Yeah.
And then we also had a question from Ann Naylor. And she was wondering, have you found any bacteria or viruses that would be harmful to amphibians? Because we've seen a lot of frogs and toads.
Yeah.
Turtles.
Yeah,
Turtles, we have a huge turtle.
that's a great question, and I think the two things I would say, I don't know the answer yet. We're looking into that because my colleague, Dr. Moore, is looking at the viruses now, and I'm sure we're gonna find some, because I've seen the turtles in there as well, and yeah, I think there's gonna be some viruses in there. Whether they're actually causing harm, that's hard to tell, but I'm sure we'll find some in there as well. Typically with amphibians, you're gonna have also fungi that might be pathogens there, and we didn't really pick up fungi in our analysis, but that's not because they're not there, but it's just because our primers in our setup is focused on bacteria and algae, and we could run different analysis for the fungi, which we haven't done. So, but I would suspect that there's gonna be some that we find in there, especially in the viruses part, I'm sure. But I'm not sure yet.
Oh, thank you. And I think
Yeah.
Kathy maybe had a
And
question.
I think you pretty much answered it. I was just gonna ask, how the health of our area, at Hill Creek and the pond compared to other areas that are nearby?
Yeah.
And I don't know if you sampled nearby areas.
I haven't sampled too many nearby areas, so we sampled some, I said the Nebraska salt marshes, because that was extreme. And we did sample some around Bellevue, where we just took some samples for some projects as well. And I would say it's not that unhealthy, because I've seen worse places, in different places. In Arizona and New Mexico, we have some samples that are a lot more in dysbiosis, so the balance is out from the sulfur cycle, and then they build up a lot of sulfur components, and then you get smells of rotten egg and stuff in your pond, and that's because it's unbalanced with the bacteria as far as the sulfur cycle. As far as I can tell, it was fairly well balanced throughout the seasons that we saw. They changed species in the seasons, but every group was represented during the seasons as well. There's a lot less bacteria in the winter, but we did find those Legionella bacteria in there that I'd be a little bit cautious about. You don't go swimming in the winter.
Yeah, it's great.
Or drink it, yeah. Yeah. I would
But that-
filter it first.
That
before you
is
drink it.
not harmful to mammals then, that it is.
Legionella, yeah, Legionella bacteria is harmful.
Yeah,
Yeah.
oh, that's right. So
Yeah.
we might harm the deer and the fox and the coyote and dogs. and
Could you guys
squirrels
speak up a
and-
little bit?
Oh,
Oh, sorry. We were just wondering if it, that Legionnaires would harm the mammals that are out there. So I guess the answer is yes.
yeah, right. I'm not sure how specific it is. I know it infects humans, and in humans causes pneumonia. I'm not sure how broad spectrum it is as far as other animals. But people must have studied that. I just haven't looked into that.
Yeah.
That's a good question. But it is a pathogen for humans, so it causes pneumonia. So, and it's typically aerosols that get in. You often get outbreaks from this in even in HVAC systems and in enclosed buildings, in the water coolers. When it's in a water cooler, it's pressed to the HVAC system. And it's typically elderly people or younger kids that get problems with it as well. And people have died from Legionnaires disease. So,
Well, we do cleanup there where people might get their hands wet. I mean, they're not breathing them, but
yes,
I was gonna say,
You have to breathe.
right.
yeah, you have to breathe in the
It's just
aerosols.
stepping in it is not. gonna-
No, that
that's
should be
what I've
fine.
done too.
Yeah. I don't want to cause any panic.
No,
You
no.
find these species in there, and
It's too late. We've already done this a couple times. We've already been deep
Right,
into this.
But it's good to keep an eye on, and that's why I want to go back this coming year and do the same sampling and see, do we get these outbreaks often, or does it come up and down, or because it wasn't there most of the year, just in that winter sample that came up, and I'm sure it's gone back down again. And so when they cycle, when the sun comes up and it's more warmer and sunnier, more species will grow, and some of these other things like Legionnaire will disappear because other things grow faster in those environments. So I'm sure in springtime, that was a minor component from all the other stuff that grows. But I'm curious if it comes back every winter, or if that cycles through, or that I haven't looked at.
Can you ask that to focus on deer and just get rid of them?
Purposely?
No, never mind.
We might have to cut that out of the recording.
you put troughs of water around. it.
Yeah.
There you go.
It's a slow death.
Oh, I know, so, yeah.
You said it's the new bacteria you found was a carbon.
It's, yeah, so it does photosynthesis and it captures carbon. So they grow with sunlight as source of energy, and then they capture carbon from different sources. They can use CO2 simply, but they can also use other carbons to store it as well. Different sugars and organic carbons. Yeah.
We're trying to get things to take
Yeah.
carbon, right? I mean,
Right.
it's
Yeah.
something that-
So, and it's something, not just here, but we're looking in general for these samples. That's also an ongoing project, is looking at methanogens, methanotrophs. Those are groups of bacteria that take up methane, or they make methane. And methane is one of those carbons that is a big greenhouse gas. It's 11 to 20 times more potent than CO2. So anything that's out of bounds there is not taking up enough methane, or is producing a lot of methane. And I haven't looked at these samples for those groups, but we started to do that with a sampling in Louisiana that we had. We're looking at the runoff from a wastewater facility, and then see in those swampy areas, how much impact is there from the wastewater facility on the bacteria growing in those environments. And then there's a group from Louisiana State looking at the plant growth and seeing how does it correlate with the plants. So there we see a big fluctuation in methanotrophs and methanogens. Basically, you see, as you go further from the water pipe, where the water comes in from the wastewater, you have more uptake of methane that occurs, and initially you have more production of methane that occurs there. So it's something we haven't looked at for this, but those will be the groups to focus on, looking at methanogens and methanotrophs. The methanotrophs grow anaerobically for the most part, so they're going to be in the deeper areas, not as much oxygen, and we would have to sample a little deeper in there as well. But I don't think we have a problem with this here necessarily. I think it does what it does. It does cycle the bacteria pretty normally, I would say.
If there's too much carbon brought into the water, does it overheat?
No, but it might change the pH, for example. It might change the balance. So it depends, several things. If it's taken up by the water itself, it could acidify the water, and it could cause the pH changes. it depends what kind of bacteria take it up. They could produce other compounds because they take up methane or other carbon sources, and they usually produce something like acetic acid or lactic acid. They produce acids that can change the pH of the water as well. So if there's an unbalance of the species, then you're typically going to see a difference in pH or a waste buildup that occurs, and then that's going to kill all the species and becomes a vicious cycle after that. So that's why I mentioned pH early on. The pH is pretty normal. I think it's around 6.5 or so in most of our measurements. So it didn't seem like anything was off balance there.
Yeah. Well, there's
Yeah.
little fishes that grow in there.
I saw a lot of fishes in there too. And typically, I haven't looked here, we're still looking at the viruses, but we often find fish pathogens in there as far as viruses or some bacteria in other places that we find. So I wouldn't be surprised if we find some fish pathogens or fish viruses in there as well. Yeah.
We're just grateful that there's fish that can live there.
Yeah.
Because it's so
It's-
covered with all that algae. I guess that's what it
Right.
is, Yeah.
Yeah. Yeah.
So,
And I think some of the algae that starts growing is possibly from the runoff that comes in from the rainwater and brings extra nutrients. So if you have too much of that, then you're gonna have these algae blooming occurring. And there's some of that in some of the seasons, but it seems to go away. It seems to settle itself. The problem with the algae blooms, there can be different problems. If it's toxic algae blooms, if it's cyanobacteria, they could produce toxins that are toxic to humans and to mammals and to deer as well. So those can cause irritation and can cause death in small animals as well. So definitely we need to watch out for that. If there's any toxic algae blooms, which I haven't seen here. The other thing is if these algae blooms occur and the algae die off, they will make a lot of debris from the algae that sinks down, and then anaerobic bacteria grow in that, and then these bacteria take out the oxygen out of the ponds, and then it becomes eutrophic ponds where nothing wants to grow and live, and the fish die off at that point. So if you see the algae continue to grow over the years and come up in blooms and going away, then you might wonder, are there too many nutrients from waste runoff coming into the ponds from these runoffs that we have here? It didn't look too bad from what I saw in 2020. But we should keep an eye on that over the coming years. Is the algae content, the blooms getting bigger over the coming years? Then there's probably extra nutrients from the runoff that comes in there as well.
Is there a good purpose to algae? Does it do helpful things?
They make oxygen, so they produce oxygen. So in general, people don't realize that, but the oxygen on the planet, depends who you ask, because the calculation, 50 to 70% of the oxygen on the planet is made by algae. So more than the trees produce. So that's why we can breathe oxygen. So there's good things for algae too. Now, as I said, if they overgrow in these ponds, these local areas, that causes problems for the ponds and causes other life to die off.
So they,
Yeah.
Oh, and then we
Well,
have
they-
another, oh, sorry, Charlie. We have another question. Actually, we have a couple of questions. The first one's from Ann Naylor, and she's asking, is there anything that we need to be doing or could do to make sure that we keep the pond healthy? And then Marilyn was asking what other creeks is Hell Creek a part of?
Well, two good questions. So what to do? As I said, I would keep monitoring it as far as nitrates and phosphates, and I'll keep monitoring bacteria and algae for you, but keep an eye on the algae, because the thing to do if that becomes a problem with algae blooms or pathogens that keep coming up is probably looking at those runoffs coming in. I think that's the main way that these things would come in. The nutrients would come in, or the pathogen would come in. And well, there's that. Looking at the runoff, that could be a bigger project to limit that runoff or to clean that runoff when it comes in of some sort. The other thing is you have a good amount of plant growth in there, and the plants will take up a lot of these nitrates and phosphates too, so that the algae don't get access to it and don't start blooming as well. So keeping a healthy plant environment around the edges of the pond, I would say, helps with balancing the nutrients as well. So it's my recommendation, but I'm not a plant person. What was the other question? again? Sorry.
What other creeks is Hell Creek a part
Oh,
of?
yeah, I'm not sure of that honestly, because I've only looked at the local area and I was wondering where it came from. I found it was like a Boys Town reservoir that it was connected to, but I'm not sure how it's connected with the other systems. I haven't looked into that.
Thank you.
Do you have a question?
Well, I was just saying that, part of the carbon and oxygen cycle, so.
Yeah.
It's not actually making anything, it's just part of transferring.
Right, right. Well, and so they do take up CO2 from the atmosphere, so they use that, or methane from the atmosphere. So they do take some of the carbon out of the atmosphere that they capture, and then they make biomass with that. So some of that is captured in there. But then there's other, when they die off, there's other bacteria that produce the CO2.
Right, I mean, it's
It's a cycle, yeah, yeah,
So you're just
right,
making the size of one component of the cycle bigger or smaller.
right, yeah.
So it doesn't necessarily add or take away. It.
Right, I mean, we want to take CO2 out of the
Yes.
atmosphere.
Same thing as
I mean,
for
the
the
net-
trees.
The net
Yeah,
loss.
it's the same as true for plants and
Yeah.
trees. They do a balancing. Just on a faster time scale than the
Right.
trees do, yeah.
Yeah.
But you make biomass in the same way.
I expected horrible news, because everything is filthy over there. Yeah.
Another question?
Well, I was gonna ask, now I forgot, of course, what I was gonna say.
That's all right.
Because I was distracted. But, oh, I know what it was. We periodically sample our soil, and I think our pH
Okay.
is high in the soil in the garden
Okay,
and in the orchard that is right next to the pond.
yeah,
Should we, I mean, you just did this, you did the water sampling just
right.
for the fun of it and
Yeah.
for the work, for the science of it.
Yeah.
But should we be periodically monitoring, like in a more thoughtful way, watering get water samples?
Yeah, it's, most people look at the water from just chemical perspective, not from bacterial perspective, because it's a lot more work to look at the bacteria. But if you can, both the soil and the water, if you can look at the microbial composition, it could tell you things that are happening, that are changing as well. Should you do it? Should you pay someone to do it? Probably not. You can just look at the chemical composition. If you're asking me to do it, I'll happily do it. So I'll keep taking samples from the water. And I was going to talk about, we can incorporate the soil with that, or some soil samples as
Oh,
well.
that'd be awesome.
Because we started looking at the soil samples from our native garden at Bellevue now as well. And we sampled 4 samples throughout the year, so the seasons as well. It's a lot more complicated going on as well, because there's a lot of stuff in the soil going on that's different from the water. And I may have looked at water samples before, but there's lots of interactions with the bacteria, wet fungi and wet plants in the soil and stuff that have an influence on how that changes the composition as well. So I may not be able to tell you exactly what's a healthy soil, what's an unhealthy soil, but if we see things come up, if you lose nitrogen fixers, then you're not going to have as much nitrogen in the soil, and you're not going to have as healthy for your plants to grow, for example, as well. So you could look at things like that again, looking at groups in the soil as well. So it's definitely something we could expand on, and it might be interesting to compare soil samples from here to the soil samples from our native garden in Bellevue. And actually, we have soil samples from I think Dr. Moore went out to 12 different places that have native gardens in Omaha and Lincoln that he went and sampled soil from there. We're doing it over several years, so we have about 2 years of data. So we'd like to get more data for these years and then see how that changes and how that stabilizes as well. But that's something we could look at too and see how does your soil microbiome compare to others, and maybe relate that to your plant growth as well.
What do you mean by native garden?
Those are plants that are all native to the area of the Midwest and
oh.
Nebraska really area.
Oh,
Yeah.
but the
Yeah.
soil isn't native, the soil's been.
Not necessarily. Well, the soil, so when we redid our area, there was just a steep hillside with just weeds and bricks in there. So they came and they leveled that, and the construction people brought in soil from the Midwest somewhere. We don't know exactly from where. So there was soil brought in. Then the soil sat bare for the whole fall and winter until the spring when we started planting stuff in there. So we do see changes in that soil happening since we planted stuff in there. So we have that year of data. And some fluctuations we understand, some we don't understand. Some things seem to go down and come back up in the next year, which we're not sure what's causing that. That's what we're doing another year to see how does it stabilize. But it definitely has changed. And the soil actually, we see a lot of acid adapted species. The soil has more acidified over the time as well. So things like that we're looking at. So, but the native plants we talk about, the native garden is native plants that we planted there. And the reason that's done is so that it will need less watering. It's more adapted to the climate, and it also grows with the or it's adapted to the insects that live here, so it's balanced out as far as pollination and things like that.
Right.
So that was the purpose of that hillside, and then some of the grass area, not having the typical just grass that you have, but having some pollination and some habitats that create. Yeah, so that was the purpose, and that we set that up. As well. Yeah.
In your walks, have you been through the garden?
No, not really. Sorry, I should. I feel guilty now. It's usually a run, so I come running by and I'm almost dying by the time I get here. So I'm hurrying to get back home at that point. So yeah, but yeah, I'll stop by somewhere definitely. Yeah.
Not
And
much to
then,
see right
No,
Yeah.
not right now. Yes.
I'm sorry, so Marilyn just had another thing that she added into the chat.
Yeah.
And she said, so basically, the Hell Creek / pond is fairly healthy in your assessment.
And yeah, what I've seen so far, and at least in that year 2020 that we measured, it looks like it's a fairly balanced system. So I wouldn't be too worried about it.
now. So are you suggesting we change the name?
I'm not sure. I don't think I have that power or authority to change the name of it. I do find it an interesting story that it's a tri-faith community around the Hell Creek watershed.
Yes.
It's kind of a balanced thing. I think it kind of works well that way. So I would keep it if it was my creek. Yeah.
And Marilyn says, keep the name, she loves it.
Yeah.
And Anne said, good news about the pond.
It
Yeah,
is.
yeah.
It truly is. I was worried,
Yeah. Yeah.
because of all of this stuff that comes down
Yeah,
there.
so there's definitely, and that, I think you're probably right, it comes from some of that runoff and stuff, and I'm not a designer or an architect, but it could be a way to capture that stuff as it comes in and then collect it regularly from getting it into the ponds. But that would take some setup to do that.
Right.
Does anyone know whether when this was a golf course, that water was essentially the same as it is now, the way it's?
No, it's been changed.
It's been, I think the
So
flow has
prior-
been changed, yeah.
Prior to, I'm Wendy Goldberg.
And
Oh,
Marlene,
Our director.
Yeah, yeah.
speak up.
Yeah, sorry. Prior to, when it was a golf course, the flow of water was actually pushed all to Boys Town. And when the zoning was approved for the development of Sterling Ridge, FEMA didn't approve continuation of pushing the water. So the sewage outage spots, or at least our corner of the woods, is the south, just south of the grove of trees. By the garden, there's a sewage outage spot up in this corner, like on the northwest side of AMI, like across the ring road, is a sewage outage spot, and then where their actual pond water is. All of those aren't owned by any of us. They are maintained and managed because they're sewage overflow spots for this whole area.
And so thanks to your help, the architects actually sent me the flow plans of this, and it's not colored in here, they had it colored and marked, but I think this whole B2 area was what's running into that, the northern pond here, so they mapped out what runs in there as far as the runoff, and it does not get mixed with sewage, that part, so it's just storm runoff from this B2 area into that north side pond. We did not sample on the other ponds, we just sampled from that north side.
So it's essentially a new construction since they moved everything around.
And to your point, there was considerable, for each of the construction site, there was a considerable amount of dirt that would be brought in prior to, they bring in those, remember when there's that huge mound of dirt out there? That took us so long to get them to move it away. What they did was they would bring in this dirt and they put it on top of where they're gonna dig the hole to help settle the dirt. They put more weighted dirt in there. And then some of that dirt just got moved around between the construction projects. But none of that dirt was like native country club dirt. It was all
Brought in.
brought in.
How long ago was that, brother? Do you know? I'm
So
just
the
curious.
construction of Temple Israel was 2013,
Okay,
was completed in 2013, broke
Okay.
ground in 2011.
okay,
The mosque was completed in 17,
okay.
and the church was completed in 19.
Yeah.
So about 2 years prior to each of those.
I think when that was brought in, by now that probably has settled and adapted its microbiome to whatever it is based on the plants that are here and stuff. I would assume.
Would you say it's reached a steady state?
Well, I think so, but that's what we're trying to measure with our experiments, like over multiple years. Because as I said in the Bellevue native garden, we now have,
It's nice to meet you. I'm
Nice
sorry.
to meet you. Yeah.
Sorry.
we have what we call year 0 is when they brought the dirt in. And now we have 2 years after that, year 1 and year 2. We'd like to get a 3rd year to see, because we see things definitely change from year 0 to year 1. And then year 2, some of the things go back to the year 0 and other things just keep going up from there. So we'd like to see is year 3 where it stabilizes, where it's a steady state. So I think it's going that way, but I don't know in general if it does. Yeah. But I would say it looks like it's at least 3 years after the soil gets brought in, going through all these seasons and changes to get that to stabilize and adapt to it. I did bring some copies of this first publication. Don't feel like you have to take this. It's very technical, it's very short. But if you want to know more about what we sampled and how we did it, feel free to grab one of these.
And so I have one more question,
Yeah.
since we were just talking about the sewage and the flow and all of that. If there was ever an issue where there was a significant amount of raw sewage
Yeah.
that ended up in either the ponds or the creek, like what would be some of the fallout from that?
So the big thing with sewage is the pathogens in there. So if it's when it's treated, but it's typically going to be raw sewage coming in there, you're going to have human and animal pathogens in there. And then you have a problem with, well, those will multiply in there. Then getting in contact with it, breathing it in, could make people sick in that process. It can cause mammals to get sick as well. So yeah, if you had, I was worried at first if there's stormwater and sewage combined in there, then you have bigger problems because it's not just the nutrients. One thing is the nutrients, you get lots of nitrous and phosphate in the sewage, and that's going to cause these algae blooms, and that's going to cause an unbalance of the microbiome in there. But also the pathogens that come with it as well. So yeah, don't mix the sewage with the stormwater. It's not a good idea. I know in normal
Write that
I still have
down.
a lot of systems that do that, but it's not a smart system to do. Especially when there's lots of stormwater coming in, and then you have your sewage being pushed back, and then you have all kinds of problems. Yeah.
I have one other question. You may know this. I should probably know this. But when was the last time that there was like a significant flood or semi-significant flood with Hell Creek? Do you know?
I don't know, honestly. Do you know? 'Cause I just read some.
In the 1960s, it says.
It's possible, yeah. I don't know for sure.
That's what the city of Omaha says.
Yeah. Because when they
That's why I have these
purposely,
ponds and other things.
that's why they have these ponds in there. So the ponds are really made, I mean, it looks pretty when you have the pond around, but they're made for reservoirs in case there's lots of stormwater coming in. It fills that up and it doesn't flood the creek up necessarily. So that's why they designed these levelings and these runoff plains, the architects did. So hopefully it's going to be a while before you get another flood of the creek too. At least in this
We
area.
could use a little moisture.
Yeah. Yeah. Any other questions? Everybody's reading the paper.
Yeah.
Yeah, good.
It's way over my head.
Dr.
Oh,
Kyndt, Dr. Kyndt,
yes.
help me understand what you mean when you say sewage. Because I'm thinking of bad raw sewage, sewage that has bad stuff in it. You know,
Yes.
like E. coli and things like that.
Yeah.
Or are you just talking about runoff from the streets and stuff?
No, so when I say sewage, I mean exactly what you say is the stuff that has bad stuff in it. And just to be clear, that is not coming into these ponds or in the creek. They have
Okay.
separate things for that. So when we talk about runoff, it's from the fields around here and from the streets. You're right. So there's going to be chemicals from the street cleaning and the street like the salting the roads. That might get in there as well, and that might change the pond composition and the microbiome some. But not the raw sewage. No, that doesn't come in there.
Yeah.
And that's, if I said if that was mixed with the runoff, then I would be more worried about.
Yeah.
Yeah. Funny looking at this owl.
Yes,
I
I
just feel
know.
like he's looking at me.
He is looking at you. Yeah.
I've never seen one of those.
Yeah, and it turns around to whoever is speaking
Okay,
a lot
okay.
of times too, which is like, okay, a little
Yeah.
too smart for me. Yeah, I think what we worry about is, preserving our animal
Yeah.
life out there.
Yeah.
I don't know how we can keep the human animal life from being so destructive out there, but
Yeah.
we worry about all of that. It sounds
Yeah.
like though we've got potential to
Yeah.
be able to make it a little better out there.
Yeah.
Do you have a recommendation for this?
See, the problem, as I said, if you do see things go wrong, it's often hard to step in and fix it. Because it is something coming in through the runoff, or it is people polluting it by throwing stuff in there. So it's not something you go in and chemically fix it, or you throw in some different bacteria in there to
Right.
fix it. You gotta attack the bigger problem if there's stuff coming in like that. So I would say if you do see things like too many nitrogen phosphates coming in from the runoff, then we got to think about where is it coming from, and can that be diverted somewhere else, or can you put some certain buffers with plants up to stop that runoff before it gets to the pond, so you can change your planting, things like that. So it becomes a bigger thing to do than just locally at the pond necessarily.
Well, where the cattails are all growing by those culverts next to the orchard is a
Yeah.
big buffer, I would think, for
Yeah.
a lot of the big pieces
Yeah.
of things that are coming.
Right, and a lot of people put that type of buffers in to get a lot of like runoff from agriculture and stuff. They try to capture them with those things. The only downside with some of that is that you plant something that grows really fast and then it starts taking over some of your other plantings. So you got to find a balance with that. That's the tricky part, I think.
Well, and I think that we're hesitant to do anything until we know what we should do, because
Yeah.
I feel like we might cause more
Yeah,
trouble.
and I know you guys are plant people and have the background, so but if you want my colleague Dr. Moore to come talk about native plants and the benefits of that, I'm sure he'll be happy to do that, because I'm a microbiologist biochemist, but I'm not a plant person, so I apologize. But he will know more about that and about which plants recommended for buffers or for native areas. And because one thing, him and Ben Boyd, I was talking about Ben as well, they had a plan together for our native area where they had things that grow in different seasons, obviously. So things come up early in spring, and those will die off, but then by that time, other native plants have come up that take over. There's always something that's green and some often blooming as well in there. And then they seeded some things that come up every year, and sometimes they have to reseed as well, but that's their domain. They had a pattern of this, and this pattern was with close to 20 different species. And this pattern was repeated several times in different directions to create diversity in the area. Because one thing he'll tell you with the native plants is you see a lot of people putting native plants and they put them row by row and they put different species. But that's not really creating the best habitat. The best habitat is to mix it in with each other and have a balanced system that way. So that's what he's going to recommend for sure.
Yeah, oh, that's good. Yeah, well, we've had several people say that they want to skim all that green stuff off the pond, and that we needed to get, more light and
It's-
air and all this.
Yeah.
It's just like, don't touch it.
Right, because it's and we have one other pond at Bellevue University, which is a pond that they have koi fish in it, and it's for presentation mode basically, and they don't like any algae in there as well, and it's a never-ending battle of taking the algae out, and they have little plants around it, so all the nutrients that come in there are just the algae is blooming every year, and they've asked us to look at it, and we're like, well, if you have more planting around it, if we can change the layout and design, then we might be able to make a change. But sometimes it's just the system, the way it's set up.
Yeah.
But yeah, definitely keep an eye on those algae blooms, I would say. And if it does become a problem, then you got to find out where the nutrients are coming from for these algae, because it's
Sample
yeah,
the drains coming in.
the drains coming into it. There's lots of runoff coming in from nitrogen phosphates. Then you're gonna have more blooms of the algae. So,
Next time there's a storm, you go out there and collect the water.
I'll do that. I'll just
Yeah.
get my waders on.
Yeah.
Get out there and do that. Yeah.
Cold.
Yeah. Breathe
And,
in a little bit.
Yeah, whenever you guys want a sample taken, or you can take it yourself and give it to me, or you can tell me and I can come and take a sample too. And we can measure, so easily with strips, we'll measure the pH, nitrate, phosphate. That's an easy measurement to do to see if that is out of the ordinary. The bacterial analysis takes some time because you gotta take it to the lab, you gotta extract the DNA, then you gotta make a library, and then you gotta sequence that library and then analyze the data. So it's a bigger project. That's what we want to do it 4 or 5 times a year. But otherwise, water analysis we can do fairly easily as well. We won't know chemicals, we won't know necessarily what organic chemicals are in there, but we can tell the total carbon and stuff in there. So
Okay.
so,
That's great.
yeah, so we can do that if you guys, whenever you guys have a need for it. we can
Yeah.
stop by. It's fairly easy for me to come.
So I don't know if anybody has a suggestion about what to name that bacteria, but it should be something trifate.
Yeah, I haven't got any suggestions yet, so we'll take it.
Infernalis.
That was on the table. Infernalis was on the table. Yeah.
Infernalis.
Yeah.
Infernal.
Oh,
From
like Hell's
Hell's
Creek.
Creek,
Oh,
That has been suggested. Yeah. I think because we sequenced it 3 times because the first time we had contamination of other bacteria still in there. So I think the first and second time that was the name of it that we had, and then we left it blank the third time we hadn't decided on it yet. So yeah, when we fully characterize it, then we'll come up with something. But, yeah, but the trifate something would be interesting too. But it's typically a Greek or Latin name, so we got to come up with some Greek or Latin
what's the
version.
word for unity?
And then we just had a couple of comments in the chat. Anne said that she would love to hear what Dr. Moore would have to say about native plants.
Yeah.
We'll
Okay.
have to get Dr. Moore here. And then Eileen said, thank you so much for sharing your research with us, which yes,
Yeah.
agreed.
Yeah.
And Anne also said, very interesting presentation. Thank you, Dr. Kyndt.
Thank you. Yeah, it was very informal. I could have brought slides and stuff, but I
This
didn't
was great.
realize. So, yeah.
This was great.
I've seen lots of slides.
Yeah, sometimes that's too much.
This was very good.
Yeah, it really is helpful. It
Yeah.
really was helpful for me personally.
Yeah,
just
I'm
not
glad
to
it
worry.
is. 'Cause,
It's reassuring.
It was.
yeah, good. I still wouldn't go swimming in it though.
No. Ann Naylor, the lady that's had so many questions,
Yeah.
is a person at church, at our church here, that puts on Earth University. So
Okay.
she has all these environmental issues that she,
Yeah.
finds speakers for, and so
Okay,
she delves deep into that. So
Yeah.
she's,
okay. Good, good.
taking over in her head what's next.
Yeah.
Yeah, exactly. So,
Yeah, and if you have groups or classes or organizations that want to learn how to do this, then we can work with them too. They can sample with us, and we can show them how to build a sequencing library and do the sequencing itself as well. It's, I know we seem over your head, but we had a summer camp this summer in June, second week of June, a high school summer camp on learning how to sequence. And they sampled soil from our native garden and some areas around Bellevue, and the kids brought the sample, they extracted DNA, they built the libraries, and they did the sequencing with some help from us. They did the sequencing in the end and got the data from it. So the data was a little over their head as far as the bacteria stuff, but they saw the whole process of how that's done. I think the biggest data thing was I had them guess how many bacteria we're going to find, and we had a raffle in the end. So, so the closest number got some prizes, but there were thousands of bacteria in those samples. Yeah, something like that, right?
Oh my
That's kind of thing.
goodness.
So yeah, we're open to teaching it to people that are interested, especially if you have groups or organizations that say we want to do our own sampling, and then we can help set that up too. So,
That's so great. Really, this is just wonderful.
yeah, happy to help.
Oh, that's great. Really, it's just wonderful to know that. And the invitation is out there for you to visit not only the garden, but the
Yeah.
orchard too. Or
Okay.
show you.
Or any of our communities.
Yes,
The doors
yes.
are always open.
Okay,
Yeah,
thank you.
that's
Yeah.
right.
I'll definitely stop by probably in the spring. Stop by the
Yeah.
garden and do what you do.
There's not much to look at. We just had Chris isn't here in the room with us, but she helped organize, I don't know if you're familiar with Hillside Solutions composting.
Yeah.
Yeah, so we had, how many square yards? 60 some square yards of compost delivered to our,
Okay.
yeah, so it looks like burial mounds out there right now.
Yeah.
So it's not beautiful right
Right,
now. So, yeah, we're very proud of it. So we haven't been out there even 3 years yet. It'll be 3 years, Mother's Day.
Yeah. Well,
So we're just now getting started.
is it all volunteers based? Then?
It is.
Okay. It's a lot of work.
It is a lot of work.
It's typical, it's a vegetable garden.
Yeah.
Everything's donated
Okay.
to local.
How much was donated?
Yeah.
How much was donated, Kathy
Yeah.
says? 2,500 pounds.
Oh wow. Yeah.
25, 35.
Yeah,
Right,
yes. To be specific, yes, so, yeah, we're really happy about that we're able to donate that much.
Yeah, absolutely.
Yeah, and then hopefully next year, Rick is the manager of the orchard, and hopefully next year we'll have fruit. Right, Rick? Yes.
Good.
Even if we have to buy it.
Yeah.
Even if you have-
Put them on there, yeah. I
This
know.
time one with a string on the.
Yeah. I know,
Yeah.
it's been
Yeah.
tenuous to start an orchard, I
Yeah.
think, but
Well,
it
it
takes
takes time.
time.
It does
Yeah,
take time.
Trees take time.
Yeah.
that's right, and then they're there for a long time, but still, yes, knock wood, yes. We've been dealing with all kinds of little pests, we're organic, and so
Yeah.
we don't,
Yeah.
the way we handle anything
Right.
that we're having trouble with is,
Yeah.
pretty hands-on.
Right.
So, yeah.
Yeah. Somewhat related to that, we had a wasp nest in our native garden this fall that came up, and there were the ground hornets that were in there. And
Okay.
so our students are sampling in those native gardens, and they're measuring the plants as they grow every year as well. And then my son, he's actually into insects, and he came over the summer. We made it his summer job to twice a week had to go through the native area with a net and collect insects, and then count how many insects we have. And then he releases them, but it's just to count do we increase the population of insects. And he actually found the nest. He's like, there's lots of wasps in here from somewhere, and we found it in the ground. And then several students got stung after that, and we're like, oh, we got to do something with
What did
this.
you do?
Nothing successful.
Uh-huh.
we didn't want to spray any pesticides in it because it's our native garden. So it came to the point that our lab manager, Johnny Farnham, he's a former Navy guy. He has all these ideas. So he came up with liquid CO2 and actually closed the holes up and pumped the CO2 in there. And
Frozen to death.
he tried to freeze into that, and it did not succeed. They came back the next day, so it wasn't sufficient.
So like,
Apparently,
like yellow jackets, is that what we're talking about?
yeah, the smaller, the ground hornets, they call them. Yeah, they're
Oh.
like yellow jackets. Yeah.
Oh, yeah,
Yeah,
so
that's what we got.
that's what we
Yeah.
had, yeah. And his
Yeah.
wife
And
was the magnet for.
yeah, and apparently they make their nest pretty deep underground, and a pretty big nest, and they're hard to treat anything. I mean, most of them will freeze over the winter, but then they might reestablish the colony the year after that. So,
You can't just like take hoses and.
Typically
water?
not, they have multiple exits on the nest as well.
Yeah.
Yeah, so it's a hard between. So, we're hoping that we'll have less of it next year. But we'll see.
Yeah, where we had them in the garden, don't no
Yeah.
longer exist because it was a mulch pile, and once
Yeah,
they
yeah.
left,